Welcome to this editor special episode of the HemOnc Pulse. I'm your host, Nicole Tucker, managing editor of Blood Cancers Today. Joining me is Dr. Paul Richardson, clinical program leader and director of clinical research at Jerome Lipper Multiple Myeloma Center at Dana-Farber Cancer Institute. Today's discussion will focus on XPO1 inhibition in multiple myeloma. Let's get a better understanding of disease management.
Okay, so I've got a cup of tea in hand. I'm ready to go.
Amazing, thank you. To kind of start at class level, can you walk us through XPO1 inhibition as a therapeutic strategy in multiple myeloma, why this pathway matters biologically, and what drugs are currently available or in development that target it?
Well, Nicole, thank you very much for the question. I think to help simplify it a little bit for the audience, because the actual science behind it is really quite important, but complex. XPO1 inhibition is really all about blocking what's called the nuclear exportin mechanism, which is the way malignant cells utilize the ability to export tumor suppressor proteins outside of the nucleus so that they're no longer functional and allow the malignant genome, as it were, to really do its dirty work. And so exportin is a mechanism that's used in cellular biology broadly, but in the malignant setting, it's utilized by cancer cells to export tumor suppressor proteins. And also, most importantly, they use this pathway to transmit signaling to the cytosol that can activate growth and resistance pathways. So as a very good analogy, actually, it was first coined by Dr. Joe McHale, who's now at the IMF, where it's a bit like the pathway is the mechanism by which parents can exercise control normally within the cell, within the home. But when this becomes malignant, basically there becomes a rave within the nucleus and the parents have left the home. And not only have they left the home, but the malignant cell utilizes this pathway to send signals to the cytosol and elsewhere, as it were, to bring more party members or party ravers to the party, as it were, to further emphasize or exemplify, I should say, the malignant phenotype genotype. So it's a very important pathway that controls a number of growth and resistance mechanisms and survival mechanisms in malignancy. And in myeloma, it's become very important, particularly as a mechanism by which 17p deleted myeloma is able to overcome drug therapy and unable to exert its classic drug resistance. And basically, it's therefore a very important pathway to disrupt in order to bring back the parents, as it were, to the home, stop the social media from bringing more party goers to the rave, and so disrupt the tumor biology accordingly. Now, in myeloma, we've realized this is very important. And the first-in-class drug that actually takes this on is, of course, selinexor, which is an already bioavailable agent that's currently FDA approved for the treatment of relapsed refractory myeloma, and is also now approved in combination with a variety of agents, but most importantly, with bortezomib.
Excellent, thank you. Great metaphor, by the way. So with that framework in mind, how does selinexor kind of distinguish itself among the XPO1 inhibitors, both in terms of its mechanism and the clinical evidence supporting it?
Well, I can speak with experience and knowledge of selinexor primarily. I would simply say that it's an orally bioavailable drug. It was originally developed on a twice-a-week schedule, which we realized was very active as a monotherapy, but was also not easy for patients to tolerate. So it's since evolved into a weekly schedule, and it's typically given with steroid and then combined with backbone agents such as bortezomib. Now, in the original STORM studies, which it was a privilege to be part of, led by my colleague Dr. Sundar Jagannath at Mount Sinai, we were able to show that selinexor given twice a week with steroids was able, in a subset of patients, around 30%, to generate meaningful responses and at the same time durable responses in highly resistant myeloma, and above all, be able to overcome resistance to almost every other treatment at that time, which was so-called triple-class refractory disease. When we took it to its next steps, which was really to combine it with other strategies, best partners appeared to be multiple. Essentially, you could combine selinexor with both proteasome inhibition, in particular bortezomib, and with carfilzomib. And then very excitingly, you could combine it also with immunomodulatory therapy, and in particular with pomalidomide, And these combination studies were led through the framework of a STOMP study, which was a multi-armed trial, very innovative, in which the selinexor dexamethasone backbone dosed on a weekly basis was partnered with various backbone agents, including the proteasome inhibitors and immunomodulators, but most importantly, more recently, CELMoDs. And so I think the strength of selinexor lies in its combinability with other drugs, and the fact that when you go to a weekly schedule, and in particular when you combine it with, for example, the proteasome inhibitor bortezomib, or for that matter, carfilzomib, it's exquisitely active. And I think the one exciting thing about the Boston trial, which was the pivotal trial that led to full FDA approval from beyond accelerated approval, which it was, again, a privilege to be part of, we were able to show in the Boston study that when we set selinexor and bortezomib and dex on a weekly schedule against a more aggressive standard of care at that time, which was the classic bortezomib dexamethasone schedule twice a week, not only were we able to show significant progression-free survival benefit and a significant trend towards survival benefit, but very interestingly, tolerability was very manageable. And one of the most interesting features was that the neuropathy of selinexor combined with bortezomib and dexamethasone was significantly less than it was seen with classical bortezomib-based therapy. So this was both a gain therapeutically for patients and also from a tolerability standpoint.
Thank you. Let's dive a little deeper. So as you just alluded to while explaining the clinical evidence, selinexor's clinical journey in myeloma has evolved significantly from early monotherapy studies like STORM to combination approaches. How would you describe the evolution of its role in practice?
I think the evolution of its role has been a very important one, because it sort of exemplifies what in myeloma therapeutics we've done over the last 20 to 30 years, which is to bring into the therapeutic arena novel agents with novel mechanisms of action targeted to overcome drug resistance. And by doing so in the disease, then move to combinability with other drugs and really bring therapeutic benefit to the clinic. With selinexor, the schedule now of weekly with aggressive, proactive approaches to antiemetic treatment, and at the same time to improving tolerability, be it combining with steroid, the use of Zyprexa at night, for example, the proactive use of thoughtful antiemetic strategies, including such innovative approaches as Virubi, which takes you to a next step in terms of antiemesis. We've been able to really maximize selinexor's tolerability and improve its efficacy. And I think the combinability with other drugs has been vital to its success. As I mentioned, bortezomib weekly has been a classic way of delivering selinexor, very effective and very well tolerated. I personally use selinexor with carfilzomib as well and find similar benefits in that setting. I think that one has to be very careful with selinexor to be proactive in symptom management, because once you fall behind in terms of its side effect profile, I found it more difficult from a patient perspective to compensate and catch up. So I tend to be ahead of it by requiring patients to come in weekly, which they're doing anyway for their backbone agent, and at the same time be aggressive with hydration, monitor serum sodium, be clear with them about the proactive nature of the antiemetic regimen and the use of Zyprexa to ensure that they don't fall behind. And it's very interesting, Nicole. There are a group of patients who tolerated selinexor remarkably well, and there are another group who can be an important majority in whom you have to be very proactive. But over the years of using selinexor now, I've become very impressed that once you find that right spot, the majority of my patients tolerate it well. And I think that one key aspect is dose. When we originally developed the drug, we were going as high as 100 milligrams, and obviously 80 and 60 and 40 have now been explored. And we've realized that you can see activity from doses as low as 40 milligrams weekly, with a sort of sweet spot being between 60 and 80 milligrams in my experience. And at the same time, sometimes you have to go higher, and patients can tolerate it. But again, my preference is to be in the 60 to 80 milligram range, with 40 used in selected patients. And I find it very interesting, Nicole, as we moved into the era of immunotherapy, selinexor's value has become, I think, even more important, because obviously, as we move into triple class, quadruple class refractory patient populations in whom, unfortunately, not only have IMiDs, PIs, and CD38-based therapies failed the patient, but we're also seeing now an increasing number of patients in whom BCMA-based strategies have failed them. The use of selinexor as a bridge, both in terms of syncopating treatment to next steps, and at the same time actually moving on to next steps has become, I think, invaluable. One very exciting aspect of selinexor's pharmacodynamic effects is that it can actually engender T-cell reconstitution, which was really somewhat unexpected, actually, but appears to be very real. So there's an ability for selinexor to assist in restoring immune function. And this is particularly important now in the era of BCMA-based therapies, where many of our patients become rapidly immune exhausted.
Amazing. Thank you. So at the recent ASH meeting, we saw new data involving selinexor-based regimens. Which readouts stood out to you the most, and what do they add to our understanding of where selinexor fits in today?
Well, Nicole, that's a lovely question. I think in the interest of time, because there were a number of presentations, a number of important discussions around selinexor across the whole of the meeting, but perhaps I should just focus on what, has been actually a major part of our work led by my colleague, Dr. Clifton Moe, which has been combining selinexor with, I think, a very exciting new class of oral agents, the CELMoDs, and in particular with mezigdomide, which is the leading CELMoD in terms of its activity. Iberdomide is the other one, and this is a great drug in itself, but is really being developed for use in first line and maintenance as well as early relapse. And early relapse is probably where it'll find its first approval. That's for iberdomide. Mezigdomide, in contrast, is very powerful and there to tackle the toughest disease in both later and second line relapse. And at the same time, we're seeing it now being pushed towards FDA approval in first relapse, which I think is very hopeful, to be fair. That being said, where it really is of interest is in the context of prior BCMA exposed patients and triple class refractory patients. And so with all of that in mind, the mezigdomide combination trial, which also incorporates dexamethasone, is now part of the STOMP study. And as I mentioned, my colleague, Dr. Clifton Moe, is leading this. And both Cliff and I have been very impressed with what we've seen so far. It's essentially showing a consistent, favorable tolerability profile at low dose between 40 and 60 milligrams of selinexor. Mezigdomide's being dosed between 0.8 and 1 milligram. And we've had some amazing results. And perhaps I can share an example. A particular patient of mine who unfortunately had cilta-cel fail him within 10 months. He'd had significant tolerability issues generated by some pulmonary toxicity from the CAR T. And he's 17p deleted high-risk myeloma. So we initiated selinexor and mezigdomide-based therapy to support him. And unfortunately, during the course of his initial phase of treatment, he ran into rhinovirus infection from his kids and at the same time got very sick from that. We were able to get him through all of that, but had a very delicate situation with his lungs because of the residual pneumonitis that he'd been left with from his CAR T. In any event, we were able to get him through all of this, and we saw literally with the use of mezigdomide and selinexor in combination and dexamethasone, stunning results. Just to give you one sort of actuarial number for him, his light chain on day one of one particular cycle was as high as 13,000, and within 10 days we cut it down to 2.5. And also extramedullary disease that had been literally coming out of his left chest wall had almost disappeared from being the size of a baseball. It had almost disappeared to being non-palpable. And that was in the course of basically a week of therapy. So this combination was really quite striking. And I think more scientifically in the poster that we presented at the meeting, in which Cliff beautifully framed all of this data, we looked at T-cell subsets and T-cell vigor, and were able to show very convincingly with a combination of mezigdomide and selinexor that it was exactly doing what we thought it would do, which was actually restore immune function. So this was really very interesting. And I think it in part explains why, for example, the patient I just shared with you responded so robustly and so quickly, because I sincerely believe we were reprogramming and resetting his immune system. Because I think whilst the small molecule effect is very real, both for mezigdomide and for selinexor, that kind of rapidity of response can only come if you're really activating your immune system as well.
Awesome. Thank you, Dr. Richardson. That's great news for XPO1 inhibition and great news for your patient as well. Where do we go from here? So, given the rapid expansion of options in relapsed refractory myeloma, including bispecifics and CAR T, like you just mentioned, so to kind of close us out, how are you thinking about sequencing selinexor in real-world practice?
Yeah, that's a great question, Nicole. I would say that, again, I'll pivot back to real patients on the trial. One of my patients on the trial, one of the lead patients on the study, had been lined up for CAR T therapy. And for various reasons, he, at the family level, decided he wanted to defer on CAR T. He was frankly a little bit concerned about some of the toxicities that he was hearing about, particularly late onset Parkinson's and chronic infections. And, obviously the real world treatment related mortality rates that are being reported are between 5 and 10%. And that's just a hard reality. And so he was actually a very well-informed patient. And he said, "But look, Paul, can I look at other options and before I go on to this?" I said, "Of course, let's look at mezigdomide and selinexor." And he had aggressive disease coming into the trial. He's since had a complete response and has now been on almost a year of therapy and doesn't want to, well, in fact, it's over a year now to be precise. And he doesn't want to move on. He wants to stay on this treatment. He is interested in CAR T in the longer term, but not right now. So in a sense, he's pre-CAR T. And then, of course, I share with you the previous example of our patient who is post-CAR T. So I think it belongs kind of every which way you need to use it. I think the fact of the matter is that there are patients in whom I've used selinexor pre-CAR T. One particular example did very well with that approach. And I'm going to now use it in salvage because unfortunately, he also enjoyed about 10 months of disease control, similar to the last gentleman I described, despite achieving MRD-negative CR after his cell-to-cell. But that being said, we're going to use a selinexor-based platform now to help get him back under control and get him into a decent remission where we can now then go to either bispecific or an alternative CAR T targeting perhaps GPRC5D. So my point of using these patient-specific examples, Nicole, is just that. I think one can think of using selinexor early in relapse, later in relapse. And the important thing is one can repeat its use with different partner drugs, especially if the tolerance of selinexor has been reasonable. And I've certainly done that and will continue to do that.
All right. Thank you so much. I really appreciate you taking the time to join us on the HemOnc Pulse, and we hope to have you back.
Nicole, it's my pleasure, and obviously really appreciate your interest in myeloma, number one. Number two, our new drugs, and really do look forward to helping you with further discussions in the future.